The next section of this presentation of publication for beginners is related to the ethical aspects of journal publishing, and particularly avoiding some of the ethical pitfalls that may arise when you're preparing your work for publication. We are fortunate that ethical issues are relatively few among submissions to our journals, as they are for most reputable biomedical journals, but there are times when things are done that are either inappropriate, just usually from a lack of understanding about appropriate standards, or sometimes they're outright fraudulent. So the types of things that we see in submissions to our journals are, first of all, inappropriate but not fraudulent manipulation of figures. I'll talk about that in some more detail. Disputes amongst authors, very difficult for the journal to sort out, and usually that's a job for your institution. Plagiarism. Data fabrication and falsification, which of course is very serious scientific misconduct. Conflicts of interest related to either the funding for the study or some affiliation that authors may have with, for example, a company that may have an interest in the outcome of the work. Duplication of data amongst various publications. Duplicate submission and publication of parts or whole papers. And then of particular concern for physiologists, animal welfare concerns, human welfare concerns, and more rarely other issues such as reviewer bias and irregularities in submission. And our purpose in explaining some of these ethical issues to you is really to give you the tools that you will avoid falling into any of these pitfalls, because it really can have a very significant impact on your career. So, one of the most common things, as I mentioned, is inappropriate figure manipulation. And as our studies increasingly use images of various types to make scientific conclusions, we're seeing more and more images in our journal pages. And so it's very important that you understand what can and cannot appropriately be done. So we see improper editing of images, we see improper grouping of images, and we see improper adjustments of images. And the bottom line is that as an author, you should not either move, remove, introduce, obscure, or enhance any specific feature within an image. In essence, the rule of thumb is that your images should appear exactly as captured in the lab. And if you make any adjustments, it should apply equally to the whole image. So here are some examples of prevalent figure manipulation issues, using a slide that was provided courtesy of Christina Bennett, the ethics manager at the American Physiological Society. So a lot of these issues, although not exclusively, arise in various types of blots or gels. So for example, splicing of gels is not appropriate. So the upper part of this section of the slide indicates the blots that were submitted to the journal, but when they were examined, they were actually comprised of several pieces spliced together. So as a reviewer, or ultimately a reader, you cannot tell whether these bands were all run on the same gel. Now, it's okay to splice blots together if you have to remove a lane that is irrelevant to your experiment. But in that case, you should clearly disclose that you have spliced the blots together by putting either a dividing line or some white space and explaining what you've done in the legend. Another issue that we see with blots is drop backgrounds. So as you can see in this panel here in the upper right-hand side of the slide, the contrast for these blots has been enhanced so significantly that you can no longer see any background material. And again, this does not allow a reader or a reviewer to be able to tell whether the lanes were all run on the same gel, which of course is important to make comparison between lanes. So do not adjust the contrast so significantly that the black background is completely obscured. And then finally, there may be issues with low resolution images, such as can be seen in the lower right part of the slide. Again, this is an issue because they are at such low resolution that you can no longer tell if these gels were spliced together. And in fact, if you had inspected this particular blot very closely, you would probably get the conclusion that they had been spliced. But with such low resolution, you cannot be sure. So the bottom line for submission of images is that they should appear as originally captured. And again, this is another example that was provided by Christina Bennett at APS. This is the image that was submitted to the journal. It's a blot with 6 conditions. The actual conditions are actually irrelevant. But when the art department reviewed the image, it appeared that there was a bright dividing line between the two halves, raising the suspicion that the actual blot that was shown had been spliced together. So the journal in question asked the authors to supply the original Western blots from which this image was obtained. And indeed, they had been spliced together. Furthermore, the bands, and these are the original bands here that were supposedly made up the original submitted image, had very little resemblance to the processed bands that were finally shown. And needless to say, this is fraudulent manipulation of a figure, and this paper was rejected.